Welcome back to the Biochemistry for Health Sciences channel. In the previous videos, we started talking about lipids. And the first lipid we talked about was the fatty acids and triglycerides. The next lipid we're going to talk about today is a class of lipids that we call amphiphilic lipids. And we'll talk about the importance of these lipids, amphiphilic lipids, in the formation of membranes in this video. So what is an amphiphilic lipid? An amphiphilic lipid is basically a lipid that has a polar hydrophilic head. We call this a head. So one part of the molecule likes water, we call that the head, and the other part of the molecule does not like water. It is non-polar or hydrophobic. Okay, so the typical types of amphiphilic lipids or surfactants, these have a lot of names, surfactants, sometimes called surfactants, or in everyday life, detergents, soaps. So there are a lot of different names given to these classes of compounds, but for the human body, we call these lipids amphiphilic lipids. So there's a tail and a head. The ones we find in our body has usually a head and two tails. So that's a tail, that's a tail. Okay, one good example, a very common amphiphilic lipid found in our body is called, the common name is called lecithin, and also sometimes called phosphatidylcholine, phosphor because there's a phosphate, choline is this, and there is a glycerol, sort of a backbone here, glycerol. And then we see one fatty acid here, and we see another fatty acid here. And these fatty acids can be anything. They could have 16 carbons, 18 carbons, they could be saturated, monounsaturated, polyunsaturated. So these are a variety of structures. So even phosphatidylcholine or lecithin, we are not talking about one chemical, we are talking about a class of chemicals that have this basic combination of choline, phosphate, glycerol, and the two fatty acids. The soaps that we talk about in everyday life is basically, you can see a long fatty acid that is attached to a small polar head. So this part here is quite polar. This is the head. Okay, and these are called soaps or detergents or surfactants. Amphipathic is the same as amphiphilic. So some medical literature might use amphipathic, same thing. Sometimes we'll use amphiphilic. We're going to use amphiphilic in this video. So this is what is special about these chemicals. The molecule has two parts. One part that likes water and one part that doesn't like water. So this chemistry is very important for the functions of these molecules in our body. So remember this. Two parts, one that likes water, one that doesn't like water, what we say amphiphilic. So not surprising that since shapes are very important for the human body, shapes, You can see amphiphilic lipids can have all kinds of shapes too, and when we have a variety of amphiphilic lipids. So lecithin is a class of compounds where again the fatty acid can be any kind. But besides lecithin, or what we call phosphatidylcholine, this is an important name to remember because we'll look at some lab tests about this. So besides lecithin, we have a variety of other amphiphilic lipids. They are called sphingolipids. That's because you can see the red part here. The red part here is a compound called sphingosine. So sphingolipids are basically lipids, amphiphilic lipids that have sphingosine. So we have ceramide, we have sphingomyelin, we have cerebrosides, we have gangliosides. Okay, these all contain sphingosine and therefore called sphingolipids. Any amphiphilic lipids that has phosphate can also be called phospholipid. So sphingomyelin is a phospholipid as well as a sphingolipid. Phosphatidylcholine is a phospholipid. It is not a sphingolipid, it doesn't have sphingosine, but it is a phospholipid. So you can see in all of these structures we have a head. Here we have a small head with two tails. Here we have a bigger head with two tails. Here we have the head again with two tails. And here we have even a larger head with two tails. Notice that one tail in sphingolipids actually comes from the sphingosine. So this tail is actually from the sphingosine. However, this tail is a fatty acid. Okay, so one of the tails of sphingolipids actually is a fatty acid. And again, the fatty acid type, saturated, unsaturated, or mono or di, or polyunsaturated, that all can vary. And of course, we mentioned about how unsaturated fatty acids give you a bent shape compared to saturated fatty acids. So you can see these classes of compounds can have a variety of shapes. Amphiphilic lipids can have a variety of shapes because the fatty acid part can be highly variable. Also important to note that this part here, the head of cerebroside, is actually a carbohydrate. That's why we call this glycolipids. So the glyco and then the lipid. Same thing with gangliosides. It's a glycolipid because this part here is carbohydrate and this part here is lipid. Okay, so some of your amphiphilic lipids are actually combinations of lipids and carbohydrates. So the question we really need to answer is, what's so special about these molecules that have parts that like water and parts that do not like water? What's so special in their chemistry that allows them to do what they do for our cells, for our body? So let's look at that, try to help answer that question. So to help answer the question, let's do this. Look at this diagram here. We have an amphiphilic lipid. Here's an amphiphilic lipid molecule. Here's another amphiphilic lipid molecule. Now, as long as the concentration, the amount of these molecules is less than what we call CMC, critical micellar concentration. As long as the concentration is less than CMC, the water will surround these molecules and they are all independent. They are all independent. Okay, they are all independent. However, once the concentration of these amphiphilic molecules increases, gets greater than CMC, then this is a very unstable system. It is unstable means too much energy. Then there's too much energy. okay, unstable, if there's too many of these amphiphilic lipids in the water, then what happens is it spontaneously starts forming these structures, what we call micelle, for example. Okay, so a micelle is a more stable structure in terms of energy, less energy. Usually when we mean more stable, we mean less energy. Okay, so we get a more stable structure in water. So why is this structure preferred when the CMC is exceeded? That's because now the water can simply face the polar heads, and the water don't have to try to hydrogen bond with its tails, which it doesn't like to, because it cannot, right? Tails are hydrophobic. So now the water molecules are on the outside, away from all the tails, which are all gathered inside the micelle. Okay, so this effect, what causes this to come together to form a micelle, this force we talked about earlier is called the hydrophobic force. Hydrophobic force. So the hydrophobic force can make molecules into certain shapes. Very, very important concept. Micelle type shapes can be formed in water because of the hydrophobic force. Okay, in a similar rationale, logic, that's why amphiphilic lipids can form membranes very well, because the water simply has to be on the outside and all the hydrophobic tails are gathered inside, just like what you see here. So membranes and micelles are low energy, stable structures. So this is a very, very important concept. We are surrounded by water. Molecules are surrounded by water, a lot of water in our body. And because of that, this water forces the formation of certain shapes. Okay, for example, the micellar shape, because of the hydrophobic force. Very important concept, because that's exactly what's happening in the structures of many important chemicals in our body. Look at DNA, for example. In DNA, all the stuff that likes water is on the outside. Your sugar and your phosphate, same thing. Your sugar and your phosphate on the outside next to water. You see? While on the inside are all the chemicals, the aromatic bases of the nucleotides that we'll talk about in the future videos. They're all on the inside, just like what you see here. Things that don't like water on the inside, things that like water are on the outside. So DNA, you can see, is all sort of a micellar structure. It's sort of a micelle in a way. So that's why DNA is very stable, because the hydrophobic force causes all the things that part of DNA that don't like water to be inside, and all the parts that like water to be on the outside. So a more stable, low energy structure. The same concept, micellar concept, we see in proteins. So all the parts of proteins that like water will be on the surface. All the parts of proteins that like water will be on the surface. Just like here, all like a micelle, the parts that like water. And all the parts that do not like water will be inside the protein. Okay, all the parts of the protein that do not like water will be inside. So again, proteins that do almost everything for our cells have a sort of a micellar structure because of the presence of water and because water causes the hydrophobic force. So the hydrophobic force, so basically water is forming shapes, is helping molecules form very important shapes, because shapes carry out functions. Protein shapes, for example, is the reason why there are functions, what the proteins do, functions. So here's another example of a biomolecule that has a micellar structure, and that is lipoprotein. Lipoproteins, very important for cardiovascular disease such as a low density lipoprotein, LDL. And once again, you see the micellar structure. You see parts of the lipoprotein that like water on the outside, such as the heads of your amphiphilic lipids or the hydroxyl part of cholesterol or part of the proteins that like water on the surface. And those parts that do not like water, such as the cholesterol triglycerides, cholesterol esters, okay, all these parts that hate water, they will be inside. They will be inside the micelle. Okay, so again, a micellar structure is seen in many large biomolecules that we have in our cells, in our body. So membranes of cells, whether it's plasma membrane or nuclear membrane, okay, all membranes of the cells are made up of amphiphilic lipids. All right, the tail part, and then the head part. So head and the tail, head and the tail. Okay, so the heads is where the outside is, and inside is the cytoplasm. Oops. Okay, let me go back. Okay, so here is the inside, the cytoplasm, and then the outside, if this was a plasma membrane. So amphiphilic chemistry is what allows membrane formation. So let's look at the some key points about membranes. We'll use the plasma membrane as an example. So this would be the outside of the cell, and this would be the cytoplasm of the cell. So first thing here, bilayer simply means the membrane has two halves, the outer half and the inner half. Bilayer, two layers. The heads are on the outside facing water, while the tails are right here. The hydrophobic, this is the oil part. Okay, the tails, this is the oil part of the membrane. As mentioned previously in previous videos, there are many carbohydrates on the surface of plasma membrane. These carbohydrates may be attached to the lipids, or the carbohydrates may be attached to various proteins. So we have this as glycoproteins, glycolipids, and transmembrane proteoglycans that we talked about under carbohydrates. So the membrane is not only lipids, there's also cholesterol, which we'll talk about later down here. Besides lipids, membranes are also very rich in proteins. All kinds of proteins found in membranes, in the plasma membrane. Because after all, this plasma membrane is facing the outside of the cell. So it's looking at the environment. So this is very important for proteins to be there and help decide what the cell is going to do. So these proteins can be all kinds of proteins, which we'll talk about later, such as sodium, potassium pumps, and other transport proteins. They could be receptors for cell signaling. Again, we'll talk about this later under cell signaling. They could be enzymes. They could be major histocompatibility proteins, and many, many other types of proteins. So one of the important things to note about membranes is its viscosity, its consistency. Okay, the membrane is sort of in between a liquid and a solid. And this viscosity is very, very critical for a lot of biological processes, including cell signaling. Okay, so Remember we mentioned how saturated fatty acids are sort of linear, and therefore you can pack them very closely to one another without much empty space. So saturated fatty acids, when you see a membrane has a lot of saturated fatty acid, that makes the membrane more solid, more rigid. While just the opposite for unsaturated fatty acids. When you have cis, you can see the bends. And because of the bends, there are more spaces, more empty spaces. So unsaturated fatty acids tend to make the membrane more liquid. Okay, now we don't want the membrane to be, sometimes membranes need to be more solid, sometimes they need to be more liquid. That depends on the environment. Okay, so it depends on the environment. So cholesterol, which is part of membranes too, cholesterol, actually helps maintain appropriate membrane fluidity. So that's important. Cholesterol is not just we have always heard bad things about cholesterol, but cholesterol is very important to health. And you can see it affects membrane fluidity, which is very critical in many biological processes, many biological processes, including cell signaling. Okay, so just to summarize, you can see the special chemistry of amphiphilic lipids by having heads and then tails. They can organize themselves into membranes, and it is membranes that make cells and organelles. So this amphiphilic chemistry of these amphiphilic lipids is the reason why we have membranes. To really understand membranes, we need to really appreciate this chemistry, and as well as the chemistry of proteins that we'll talk about in future videos. So that is all for now. Until the next video, goodbye, and we will see you next time.